Welcome to the first lecture unit in week 2. Isabel has explained that you can use microorganisms to produce chemicals instead of using a chemical catalyst. I will now tell you more about these microorganisms and what their function is in nature. Microorganisms have been around for billions of years and can be found in many places on Earth, in many different forms and in very different conditions. For example, if you would look at your skin through a microscope, you would find all kinds of microorganisms, but also in places where you would think life is impossible, like at the bottom of the ocean, in so-called black smokers, where the temperature is more than 100 centigrades, or deep in mines in acid mine water at pH 1, or in other extreme conditions, like in freezing water, or alkaline environments such as soda lakes. So, what is the role of these microorganisms? We do know that the energy of the sun is used to convert carbon dioxide and water into organic compounds and oxygen using photosynthesis. Now, if there would be no microorganisms, we would sit in a very big pile of organic material. And the main function of our microorganisms is to create element cycles to prevent such big piles, where they consume the organic compounds and oxygen from photosynthesis and convert them back to carbon dioxide and water, creating a carbon cycle. And in that way, they also make photosynthesis possible again. And microorganisms also do that for other atoms containing, molecules containing other atoms like sulfur, nitrogen, iron. And because of their role in the global element cycles, microorganisms convert materials occurring in nature, natural feedstocks, and given the enormous diversity of organisms, they do these conversions under very different conditions, and it is obvious that microorganisms are the masters of sustainable production. Now, let's have a closer look at these microorganisms. They do come in different shapes, sizes, but as you can see, they are small, in the order of micrometers. In addition, they share a very distinctive property. They do divide to propagate themselves with doubling times in the order of 1 hour, very short. And when they divide, the DNA of the cell is copied and divided over 2 cells. And you should recognize that the DNA essentially dictates the properties of the organisms. And copying the DNA molecule is, in addition, not an easy task. With typically 10 to the 7 base pairs per DNA molecule, mistakes are bound to happen. A copying mistake is called a mutation. But such mutations can also be induced by external sources like UV light from the sun or oxygen radicals which are around in nature. These mutations can be lethal and kill the organism, or they don't have any effect at all, and the organism just behaves normal. But sometimes a mutation can improve the properties of an organism, resulting in what we call a competitive advantage. And this can happen, for instance, when a mutated organism acquires a better performing protein for the substrate uptake. It will eat the available substrate faster, and therefore it will duplicate faster, and therefore it will outcompete the other organisms. which are deprived of the substrate. This combination of competition and selection we call evolution. And this unique evolutionary microbial mechanism, division, mutation, competition, and selection, causes that favorable properties are preserved in new generations and will be improved by an ongoing mutation, etc. On the other hand, we can select from nature. And selection and evolution from the microbial treasure trove provide us with unique possibilities to shape our sustainable chemical production. What we can do is that we can select suitable microorganisms from nature, or we can use evolution experiments in the lab to improve the properties of these organisms even more. And of course we can do both. I will show some examples. Selection from nature is a very powerful method to obtain unique and suitable microorganisms. It has been used in Delft by Professor Mark van Loostrecht and myself with very powerful results. For example, it is used to select for biofilm forming organisms on sand particles by applying a high washout rate, which removes non-attaching microorganisms, making all food available to organisms which attach to the sand. And this was the basis for a new biofilm reactor technology. An application of selection principles on other microorganisms has led to many new microbial processes from the TU Delft in the past 20 years. Now, another example is now to use evolution in the lab. And it comes from our department member, Professor Jaak Pronk. A yeast was modified, an ethanol-producing yeast was modified by adding 2 enzymes which were isolated from elephant dung, and which transformed the cell into an organism which uses a second-generation feedstock, the wood sugar xylose, and convert that xylose into ethanol. However, the constructed organism produced ethanol at a very unsatisfying rate. Knowing the possibilities of evolution, Professor Pronk put the selective pressure for xylose consumption into the lab. And in 4 months, the organism showed significant improvement in ethanol production rate. And of course, improvements are still possible for this organism using this technique of evolution. Now, besides increased growth rate and production rate, evolution also allows to improve other relevant properties for a production process, such as a product tolerance. An example from Delft, also by the group of Professor Pronk, is the improved butanol or acetic acid tolerance of the Baker's yeast Saccharomyces cerevisiae in the production of ethanol or butanol. Now, to wrap it all up, we do have limitless possibilities if we go to nature to find our microorganisms. We can improve these microorganisms and turn them into feasible production units which convert sustainable feedstocks taken from nature into relevant commercial products.